Hello again, embryology students. We're on video number 7 now of our series on embryology, and let's get right to it here. Today we're going to just talk about differentiation of the mesoderm. And the mesoderm splits into a whole bunch of stuff, which gives rise to a whole bunch of stuff. Fairly complex topic. We'll keep this one short. We'll get into the differentiation of the paraxial mesoderm in the next video. Alright, so where have we been? This is pretty much what we've built over the passage of time here. So gastrulation is done. We've built the primitive streak has been built, and it's actually starting to regress at this point. The trilaminar disc is complete. We have the 3 layers. Remember, the epiblast turned into the ectoderm. Hypoblast turned into the endoderm, and we have mesoderm. I didn't color this, but mesoderm is all in white here between the ectoderm and endoderm. We've built a nodal cord, or we're in the process of, this shows it complete, but we're actually in the process of starting a nodal cord, or a neural tube to form. So neural crest cells will be forming later. So that's where we are. We haven't talked about mesoderm differentiation, and that's a very, very important topic. So that's what this video is going to be about. So mesoderm quickly differentiates about day 17 of week 3. Mesenchymal cells remember, the mesoderm is not made up of epithelial cells. It's made up of mesenchymal cells, which can morph into everything, anything they want. So they start grouping together into 3 regions. Paraxial region, which is called the paraxial mesoderm. The intermediate mesoderm is really next, moving away from the midline, and then the lateral plate mesoderm. Okay, these are all paired structures. Let's take a look at them. So here's that view from cranial to caudal. Technically, that's another thing these books are driving me crazy with. Technically, if you're looking cranial to caudal, that would be an axial view. Some call it a cross-sectional view. Some authors call this really looking from anterior to posterior, and they've taken into account folding. Anterior-posterior, that would be a horizontal view. That's hat. Horizontal. I'm sorry, not hat. That's a coronal view or a horizontal view. So some authors call this a horizontal view. Very confusing. I probably, when I teach this for real in a couple of weeks, we're going to start my first real lecture series on this. I'll probably just say cranial to caudal view just to keep things under control. But anyway, here's that view, axial view. And we can see, just like the other one, there's the ectoderm, endoderm is here, mesoderm is in the middle. But now we have formed 3 regions. These cells have clumped together. In fact, they've turned into epithelial cells, as we'll see. And we have right next to the nodal cord, which is maybe a little bit bigger than it should be. Neural plate is just starting to form here. We've formed a paraxial mesoderm on each side. Then we have a region of cells that have, all of these are expressing different genes. That's why it gives it that's why it's called these divisions, because each of these express different genes. When I mean express different genes, that means chromosomes are turned on and transcribing mRNA, and mRNA is flying into the ribosome and making different proteins, whether they be growth factors or whatever, hormones. We can just say they're expressing. That means they're building stuff because they're turned on. So intermediate mesoderm is here, and then we have lateral plate mesoderm here. So that's where we're going. This is going to split here. This would be the secondary yolk sac, this region here. This is kind of the wall of the yolk sac. The amniotic cavity would be this whole thing here. The amnion would be this tissue here. What else do we need to say? Somatic extraembryonic mesoderm is here. Splanchnic extraembryonic mesoderm would be here. Okay, I think that's about all I need to say about that. So there are some other events going on simultaneously. Now, gastrulation, we said, has been completed. And as I kind of said already, primitive streak has already fully formed, but it's regressing. Remember, it's starting to shrink away. As it shrinks away, the nodal cord is elongating. Neurulation is just beginning, just starting to form a neural plate right here. We talked about that in the last videos. I could have colored that in, but see, it's starting to bend. We're getting some neural folds right here and here. That's kind of where we're at. So what causes this differentiation? It's another, the simple answer is, it's more complicated than this, but simple answer is it's another BMP story. So BMP, bone morphogenetic protein, it's really BMP4 we're talking about mainly here, but I'm just going to keep it simple and call it bone morphogenetic protein. This is important in spine surgery, right? This is controversy not too long about recombinant human bone morphogenetic protein 2 is used to ensure solid osseous fusion, but there's some problems. You can see another YouTube video on that. It's the same BMP. It's a member of the transforming growth factor beta family or superfamily. And it seems, according to research, that there's a BMP gradient expressed within these cells, meaning that some cells, actually the ones more toward the midline, express only a tiny bit, if any, of BMP, while the ones farther out, the lateral region, express tons of BMP. Remember, the ectoderm had a BMP story as well, which we covered before. Okay, this BMP is originally secreted from the ectoderm cells above, and it trickles down into the mesoderm cells, which are mesenchymal, and it causes some of them to turn on their own BMP genes and other genes, although some of them don't, as we'll see in a minute. So how does this BMP gradient work specifically? In the midline, like where the nodal cord is, the kind of the folding point of the little embryo, very low levels, those embryonic those mesenchymal mesoderm cells express very low levels of BMP. And cells that secrete low levels of BMP are called paraxial mesoderm cells because they're going to differentiate. We'll talk about that in the next video. They differentiate like crazy. Somatomeres, somatomeres, somites, somatomeres and somites, they differentiate too. But we'll talk about that when the time comes. The lateralmost mesoderm cells express super high levels, almost the same as their parents above. So those become lateral plate mesoderm. And the ones in between, they express just medium amounts of BMP. So only some of those genes were turned on, and those are called, those cells were given the name intermediate mesoderm cells. Okay, back to our little picture again. Nodal cord, this is in the midline. We're looking from cranial to caudal, kind of an axial view for purists, I guess. And paraxial mesoderm is in blue here. These cells, if you took a tissue sample of this, there's not a whole lot of BMP being made here. If you took a tissue sample here, there's medium levels of BMP being made, so medium genes are turned on. If you take a sample from out here, lots of BMP genes are turned on, so this guy's cranking out BMP like crazy. Simple as that. Well, not quite as simple, there's more to it, and we don't even understand the whole story. Remember, all of this stuff regarding what genes are turned on, almost all of this research comes from animals, so we're not even 100% sure if it holds true for humans or not. Okay, let's talk about the paraxial mesoderm. It's also called, some authors, like my new favorite one now, it's got to be Carlson. It's the deepest storyteller. Segmental plate mesoderm, or segmental plate mesoderm, it's sometimes called. Again, it forms on both sides. We know that. So it's paired. What do you mean paired? So it's paired. There's a pair of them. There's one pair. There's one. There's the other. This is a pair. Remember, this is just one slice of the axial view. But these would go all the way down. They go from the cranial region to the occipital to the cervical, through the trunk region. Another side note, confusing the embryology authors. They just say trunk. When they mean trunk, they mean thoracic region, lumbar region, sacral region, and coccygeal region. Okay. Alright, so and just kind of like everything else is formed, these cells they start out as endodermal cells or even epiblast cells that migrate through the primitive streak. They morph into mesenchymal cells, and they form the paraxial mesoderm. The only difference is the ones that form in the sacral and coccygeal region, the primitive streak doesn't go that far, but there is a tail bud that develops down there, and cells move through the tail bud, morph into mesenchymal cells, and become sacral and coccygeal paraxial mesoderm. Okay, so we already know this. There's the paraxial mesoderm expressing low levels of BMP. So the question is, if high levels of BMP are being stimulated up here, how come this doesn't, and high levels are being cranked out of the ectoderm here, so all of these tissues are being exposed to pretty high levels of BMP. How come these don't express a lot of BMP? Why aren't more BMP genes being turned on? Research says it's because of a protein, another gene that's turned on in these cells. A lot of genes are turned on in these cells called noggin. Noggin has been found to block the effects of environmental BMP. It's as simple as that. So will these genes out here, or will the lateral plate mesoderm, do you think they're expressing a lot of noggin? No, those noggin genes are not turned on, therefore they express high levels of BMP. Here may be medium amounts of noggin genes. There's some more story we'll talk about here that we know. But so bottom line, noggin is a blocker of BMP. Also, paraxial mesoderm is very important for somitogenesis to occur. Some people call that somitogenesis. So after the paraxial mesoderm is formed, it's going to segment into blocks of epithelial tissue. Super important. And those blocks are going to segment even further. We're going to talk about that one in the next video. I think. Stay tuned to the end. Maybe I'll couple these together depending how fast these go. So the paraxial mesoderm will actually turn back into an epithelial tissue. Remember, because mesoderm is mesenchymal tissue. So we'll talk about that though. These blocks are called somitomeres up in the cervical region, and the rest of them are called somites. But we'll get to that. Okay, so the next region that forms is this region lateral to the paraxial mesoderm. So this is called intermediate mesoderm. That one occurs a little bit later. These occur on about day 16. This clumps together. Same with here, about day 16. This one's a little bit slower, about day 17. That one forms last. Again, it forms on each side, so it's paired. Just medial to lateral plate mesoderm, we said that. This one's a little different because it doesn't form in the cranial region. It only forms in the cervical, and the trunk region. Remember, I said the trunk was everything else. Thoracic, lumbar, sacral, coccygeal. So both authors both confirmed that, double confirmed. So nothing in the cranial region. Not true for paraxial mesoderm or lateral plate mesoderm. They form in the cranial region, cervical region, and trunk. Okay, this doesn't segment, so there's no segmentation. Now, we haven't talked about that, but again, the paraxial mesoderm will turn into segments or blocks of epithelial tissue. This one doesn't. Here it is again in green. Always showed you that. How does it form? This one's still unclear. Now we know it expresses just medium levels of BMP, but there's a little more to the story here. For whatever reason, they're able to get a better story on this. So there's two gene products. We know BMP is secreted from the ectoderm above, but another one called activin is actually secreted from the paraxial mesoderm. So there's activin genes turned on in the paraxial mesoderm that was just created. And this activin protein and the BMP, they soak in to the future intermediate mesoderm cells, and they're able to turn on three genes, PAX2, HOX4, and HOX11. And if you take a tissue sample right here and you check to see which genes are turned on and which gene products are working, unlike the paraxial and lateral plate mesoderm, you'll find the PAX2, HOX4, HOX11 genes on and gene products are released. And therefore you go, okay, that's intermediate mesoderm, and that's exactly how they can tell it's intermediate mesoderm. Pretty good with that. It's not that hard, I don't think. What is the, we'll save the story on what does the, I didn't say what the paraxial mesoderm will become. Most of the axial skeleton and ribs, amongst other things, but we'll save that because I'm going to really get into that story. But what does the intermediate mesoderm become in general? Basically, the urinary tract and the genital tract, or genital system. So therefore we call it the uro, which means kidneys and ureters. Genital is the ovaries in females, the testes in males. So the urogenital system is what forms from this intermediate mesoderm, so it's very important. In fact, a little deeper, the cells, the intermediate mesoderm in the cervical region very early differentiate into something called the pronephros, which is the early kidney. The lateral portion of the intermediate mesoderm in that region differentiate into something called the pronephrotic duct, which is super important in forming, basically kind of orchestrating the formation of the urogenital system. So we'll get into that when we talk about formation of the kidney. But that's a little precursor. I don't put stars very often on these slides. My former students will be laughing because these stars accumulate as time goes by. Sometimes there'll be like 10 of them here. I try to put a star every time I write a test question. But these are important. Boards, chiropractic boards, medical boards, my tests, all professors' tests. One way to test students is to ask you what is formed from this tissue. So not only do you need to know what tissue is formed, you need to know who cares about it, what does it turn into. So intermediate mesoderm turns into the precursors of the urogenital system. So otherwise the kidney, the bladder, the ovaries, the testes, things like that. But we'll get to that more deeply. But that's how you study for this stuff. Don't go too crazy into all these genes. I don't know. I like to be thorough. I like to understand everything. So some of these gene products will be tested on, but I wouldn't worry about those now. But if you want to start learning stuff universally tested upon, you need to learn what these regions of the embryo form and become ultimately, because all professors and teachers are going to test you on that. Okay, let's get to the lateral plate mesoderm. This is also called the lateral embryonic mesoderm by Moore, which is a Board of Chiropractic Examiners book. And I got some emails. What does that mean, a Board of Chiropractic Examiners book? They don't write the book, but they write questions. Teachers from all over the country every year have to write questions for the National Board of Chiropractic Examiners. And those questions have to come from a list of books that are on the National Board of Chiropractic Examiners website. That's why I call them board books. All right, so lateral plate mesoderm. Some people just call it lateral mesoderm. It starts forming at the same time paraxial mesoderm does. Forms lateral to the intermediate mesoderm, medial to the paraxial mesoderm. It's the farthest away. Oh, I'm sorry. What am I talking about? It forms lateral to the lateral plate mesoderm and the paraxial mesoderm, Pictures are always better, right? So it's furthest away from the midline, secreting BMP like crazy, we know. And it doesn't segment. So the intermediate and lateral plate mesoderm do not segment. They don't turn into blocks of epithelial tissue like the paraxial mesoderm does. Let's take a look at it. See, it's lateral, right? This is the midline. So this is a little bit lateral, medium lateral, far lateral, you could say. Lateral plate mesoderm. This one's a little tricky. We got a little more to talk about that. How does it form? We know already. Ectoderm releases BMP. It soaks into the mesoderm below, and there's no noggin genes turned on here, so there's nothing to stop it from expressing very high levels of BMP. So it expresses high levels of BMP, which is its main identifying factor, based on research at the time of this YouTube video, which is, what is the date today? It's a little after 4th of July, so it's July 2019. Lateral plate mesoderm can be divided into an anterior. So this mesoderm is going to split up into some parts. There's some non-physical divisions, which means you can't look at it with a microscope, even an electron microscope, and tell. But there is an anterior and posterior lateral plate mesoderm, which can only be found by taking samples of the tissue and seeing what's being secreted. The dividing line between the anterior and posterior divisions is about the 5th somite, so we'll get to those somites again in a little bit. Again, it's not a physical, it's a biochemical division. Should have put that in red, based on what genes are turned on. And this region is not, we're going to talk about how this, toward the lateral part of the lateral plate mesoderm, it's going to actually split. Cavities are going to develop, it's going to split into 2 layers. But part of the lateral plate mesoderm doesn't split, and we're talking about the part that doesn't split here. What does it form? So who cares? Why did I even mention this? Well, because it forms some stuff. So the anterior division is going to help form the heart. It has some progenitor cells, cardiac progenitor cells, which will help. We'll talk about what really forms the heart here in a second. Also, the dorsolateral neck muscles come out of this region. So there's our first muscular tissue that's formed from this mesoderm. Posterior division actually helps form the upper and lower extremities. It adds something to the buds that will form. We'll talk about those later when we talk about forming the extremities. Okay, and again, this is the part of the lateral plate mesoderm does not split, which we're going to talk about in a minute. This is always present. So let's talk about splitting. So the first thing with the splitting story is the lateral portion of the lateral plate mesoderm gets some little cavities that develop, little cystic cavities. What is a cyst called? It's too easy to call it. Cyst, just a cyst, right? In embryology, it's called a coelom. So we get these coelomic spaces. Coelom just means, when you see this word, it just means cavity, or like a little cave. So we get these little caves or little spaces developed in the lateral portion of the lateral plate mesoderm. I'll show you a picture in a minute. These are called coelomic spaces. Sometimes Carlson calls them coelomic vesicles. Here's a pronunciation guide. C-lom, like Tom. Coelom, ick. Coelomic. Coelomic. It forms, interestingly, we'll talk about the cardiogenic mesoderm in a minute. It actually forms in that as well, these spaces, so it goes all the way up into that region. So here's a little cartoon, that same one we've been using. Here's lateral plate mesoderm. Here's the medial portion of lateral plate mesoderm. That's where the anterior and posterior part of it can be differentiated. Nothing happens here, but out here, this lateral half, you get these little caves or these little cysts developing. Those are called coelomic spaces. And guess what's going to happen? They're going to coalesce together. They're going to come together and form one big cavity is what happens. I can use my Beatles thing again. Come Together, a great song. by the Beatles. Is that the name of the album too, I believe? Anyway, so coelomic spaces will come together with the passage of time. These spaces, they coalesce is the official word. Coalesce means a coming together or morphing together into one big space. New space is called the intraembryonic coelom. Intraembryonic coelom. One more time. Intraembryonic coelom. What does that mean? It means it's inside the embryonic tissue. It's not outside of it. And it's a cave. It should also be noted, we'll talk about 2 videos from now, is the folding process of the embryo has started right now, because these cavities form as we get some lateral bending, some lateral folding of the embryo. But we'll talk about that later. So now those little cystic cavities are gone, and we got 2 big, or 1 pair of these giant intraembryonic coeloms. Okay, what's going to happen next? I bet you can guess. These cavities are going to get bigger and bigger, and pretty soon, what's this cavity right here for you students who've been following along? Well, it says right over there, chorionic cavity. So that's the extraembryonic coelom, or coelom, is out here, aka chorionic cavity. So for a short time before folding occurs, we're going to actually join these 2 cavities. That's where we're going. So important cavities will form from the intraembryonic coelom. So this is down the road. So this is the 2nd month. We're talking, week 8 or so. But these cavities right here and here, they're going to help create some really important structures. So we got the pericardial cavity around the heart. When we get my 5th quarter students who've made it through my pathology classes, they know cardiac tamponade can occur. You can die from bleed inside the pericardial cavity. You can get a atelectasis, a collapsing of the lungs from a fluid buildup in the pleural cavity. So that's another important cavity that surrounds the parietal and visceral pleura, or surrounds. It's in between the parietal and visceral. This is in between the fibrous pericardium and serous pericardium, or the epicardium really, which is, well, I won't get into that now. I'm going to digress. And then the peritoneal cavity, or peritoneal cavity, apples, tomatoes, however you like to say that. That's also, so these are super clinically important. We'll talk about these a lot in the pathology that can occur in here when we get in 5th quarter with me, with gastrointestinal pathology and cardiovascular and pulmonary pathology. We'll talk about all this stuff. So we're going to get to see how it's formed right now. Okay, so as I said before in the picture, the intraembryonic coelom rips its way, that should be coelom, rips its way through the lateral portion of the lateral plate, as I said, and now we have a temporary communication between the intraembryonic coelom and the extraembryonic coelom, or a.k.a. the chorionic cavity. So more important than that, we've just created 2 new layers. That is really important. So we're going to expand on that. But again, remember, this communication is short-lived, because as the folding occurs, we're going to reestablish these cavities on their own. Intraembryonic and extraembryonic coelom will be on their own, but we'll get to that. Before I talk about these 2 new layers of tissue, let's do a flashback. This is back to gastrulation, right? There's a bilaminar disc. This is a kind of a side view. There's the amniotic cavity, secondary yolk sac, primary yolk sac is getting pinched off here. Remember, we had a solid block of mesoderm which was split, a cavity, just like the same cavity story started with little cysts and they coalesced into this giant cavern that is now surrounded. Almost all of the embryo is right here, and of its yolk sacs is right here. And this is called the chorionic cavity, or extraembryonic coelom. Extraembryonic coelom. That forms, it's going to encircle the entire fetus and its yolk sacs, except for connecting stalk, which will be right about here. This will come in a little bit more here. But when this split, just like when our lateral plate mesoderm split, it created two new layers. When this mesoderm split, it created two layers. So we have a somatic mesoderm, but you got to say really extraembryonic somatic mesoderm, because the one lateral plate mesoderm is, there's going to be an intraembryonic somatic mesoderm we'll see in a minute. But here's the extraembryonic somatic mesoderm, and then the layer left here kind of around the yolk sac wall is called the splanchnic mesoderm. Splanchnic mesoderm. But really it should be the extraembryonic splanchnic mesoderm, because there's going to be intraembryonic splanchnic mesoderm in a minute. Okay, everybody good with that? All right, let's look at the split. So there's that picture that we'll be looking at. I kind of blew it up so we can see what happened. There's our paraxial mesoderm, intermittent mesoderm. Here's our lateral plate mesoderm, and not all of it, but the lateral part, we've developed those cavities, and they split, and they communicated with the extraembryonic coelom. This was the intraembryonic coelom. They're communicating, but look what happens now. Now we form two new pieces of tissue. So we can't call this lateral plate upper lateral plate mesoderm and lower lateral plate mesoderm. Can't do that. We had to give them two names. Because they morph into our friends, there's the extraembryonic splanchnic. That's wrong. That's somatic mesoderm, right? This is splanchnic mesoderm here. No, that's right. Sorry. Yeah, that's splanchnic here. That's yolk sac wall. That's extraembryonic somatic mesoderm. So because they connect with these things, let's just call them the intraembryonic somatic mesoderm. Make sense? So the intraembryonic somatic mesoderm morphs into the extraembryonic somatic mesoderm. Great. So guess what this one's called? Well, this is called the splanchnic, extraembryonic splanchnic mesoderm, which kind of surrounds the yolk sac. I see what confused me. This is cartoon. There shouldn't be space right here. They just kind of pulled the layers apart so you could see this better. These should be right together. The only hole is right here. There's the extraembryonic coelom, or the chorionic cavity. So if this is extraembryonic splanchnic mesoderm, let's call this, how about extraembryonic intraembryonic splanchnic mesoderm. And that's pretty much what happens. They change the names a little bit, but one of the a.k.a.s is exactly what I said. But let's officially look at the slide now. There's two new layers have been formed. They communicate with the extraembryonic, or they're just part of that communication between the extraembryonic and intraembryonic coeloms. But when those split, we create two new layers, as everything I already said. So the two layers are the somatic lateral plate mesoderm and the splanchnic lateral plate mesoderm. Their a.k.a.s, we'll see the a.k.a.s is what I just said in the last second ago. There are one important thing. This one is next to the endoderm. Splanchnic mesoderm is next to the endoderm. The combination of these two, it's better to look at the picture probably, but the combination of ectoderm and somatic mesoderm, and I'm just going to call this somatic mesoderm for now, the combination makes something called the somatopleur, and that's going to be important when we talk about folding. Okay, the combination of the endoderm plus the splanchnic mesoderm, that's going to make something called the splanchnopleur, right? So somatopleur, splanchnopleur. This is just double tissue. Let's look at a picture I think will help. Now this is a busy picture, but here's that same picture we've been using. So here's our somatic lateral plate mesoderm is here. You could also call it the somatic intraembryonic mesoderm if you wanted. I'll show there's a lot of a.k.a.s. Authors are all over the plate in this. Here's the splanchnic lateral plate mesoderm here. Notice how again it morphs and connects with the somatic extraembryonic mesoderm. This splanchnic lateral plate mesoderm connects to the splanchnic extraembryonic mesoderm. Here's what I wanted to show you about folding. We can combine two layers. So here is the ectoderm, the edge of the ectoderm layer. There's the amnion right there. But these two layers form what's called the somatopleur. These two layers, there's the yolks, or there's the endoderm, and it'll be the yolk sac wall out here. But if you put these two layers together, that's called the splanchnopleur. Okay, and we'll come back to that concept in the future. Chorionic cavity or extraembryonic coelom is here. Intraembryonic coelom is still right in here. Okay, everybody with me so far? Play it back. I know this is getting kind of complex here. All right, a.k.a. city. Oh my God. There's the intraembryonic somatic intraembryonic mesoderm. You could say intraembryonic somatomesoderm is a strong a.k.a. for it. Some books, like the Indian from India, the two authors, the Singhs, call it the somatopleuric mesoderm. It means the same thing. Splanchnopluric mesoderm, another strong a.k.a. for students in that part of the world. For us, we can call it somatic lateral plate mesoderm, or parietal Moore's, the Chiropractic Board of Chiropractic Examiners book, calls it parietal mesoderm. Moore he really goes against the grain and doesn't follow what all the other authors say. Usually it drives me crazy. But he calls it parietal mesoderm, and there's a reason for that, but I won't get into that. There's a and Moore also calls it visceral mesoderm, as a.k.a. for splanchnic mesoderm. Some call it parietal mesoderm. So, drives you crazy. Makes your hair stand up when these authors can't get on the same page. When we talked about gastritis in fifth quarter, same thing. They have these big, huge conferences, and they still can't come together and narrow these AKs down. Drives students absolutely crazy because you don't know which ones are going to be on boards. So for us, parietal mesoderm could show up because Moore's is a board book. So Moore also says somatic lateral plate mesoderm, luckily. So I'm trying, these videos are geared for chiropractors, more chiropractic students, with regard to these AKs, to prepare them for boards. After folding, what do they form? So who cares about these dumb things? Who cares about these? Well, they form stuff, so they're important for folding. And after folding, the somatic lateral plate mesoderm is going to make basically the lateral and ventral body wall. So that's all the flat muscles, rectus abdominis, and the external oblique, internal oblique, transversus abdominis. That's the lateral and ventral body wall. Right? Not the ribs, though. It doesn't include the ribs. Ribs are from what? Oh, we haven't talked about that yet. That's from paraxial mesoderm, is going to turn into the vertebral bodies and ribs. It also helps form the limb buds. We'll talk about those limb buds in future videos. What about the somatic mesoderm, or the intraembryonic somatic mesoderm, or the somatic lateral plate mesoderm? What does that form? Mesentery. What is mesentery? It's a weird thing. Side note here. Why do they put embryology in first quarter in medical school and chiropractic school when these are second quarter structures? Probably not the smartest thing to do in the world out there, all you deans, because students aren't going to understand it. They haven't had it yet. They don't have to study it yet. But mesentery attaches to the intestine. It anchors it to the posterior abdominal wall. So we'll study that at our school and even in medical school. That's a second quarter or second semester structure. Digestive tract walls. So the intestines, basically, it makes up the intestines. All right, so very important stuff. There's a rare star, so you can start studying this stuff right now. Lateral folding. So once lateral plate, as I said before, once lateral plate mesoderm starts differentiating into its layers, classically marks the spot where the embryo will begin the lateral folding process. We're going to talk about that later. Okay, almost done. Let's finish up some other mesoderm differentiation. And there's even more than this, some blood vessels and things like that. I'm not going to get into that right now. We'll wait till we get to the individual areas for blood vessels. So we kind of mentioned this. There's a caudal expansion of the mesoderm because the mesoderm is growing, right? So we get some folding in the median plane or in the mid-sagittal plane. There's also a folding. There's a lateral folding, and there's an anterior. I guess that would be an anterior folding. But mesoderm, in order for that to occur, mesoderm is growing. Or why does that folding occurs? Because the mesoderm is getting longer and longer and longer. In the caudal region of the embryo, we know what causes it. It's FGF8. So the expression of fibroblast growth factor 8, those genes are turned on big time in the mesodermal tissue caudally, and that helps them split by mitosis, and they get longer and longer. This FGF8 is also important because you don't want somites forming in the paraxial mesoderm down that way. They form from cranial to caudal direction, and so FGF8 inhibits somites from forming where they're not supposed to form. You get some horrible birth defects if there's something wrong with your FGF8 gene. So we'll leave it at that for now. This is a review slide. Remember, our mesoderm isn't everywhere. We have some places where the ectoderm and endoderm are pinched together, like the precordial plate, oropharyngeal membrane region. There's no mesoderm there. They pinch together and it can't get there. It can't fill in there. Cloacal membrane is the same thing. Precordial plate will become the mouth. This will become the anus. The nodal cord in the midline, it can't get there. That runs between the ectoderm and endoderm, so no nodal cord is exactly in the midline. You don't have that. Okay, the mesoderm has been kicked out of these regions, so really the disc is only bilaminar in these regions. That's a good test question. All right, we need to talk about this card. We'll talk more about this when we get to the formation of the heart. Another second quarter structure, right? Why is embryology talking about it when we're in first quarter? But what do I know? In the third week, we talked about, let's back up. We talked about precordial plate was one of the first streams of mesenchymal tissue streaming between the ectoderm and endoderm in the midline. Then we got another stream of tissue, stream of cells right in the midline. This is a third stream of tissue in the midline. And so these special epiblast cells, they're called precardiac cells, Carlson calls them, just like when we created generic mesoderm. Epiblast cells migrate through the primitive streak and primitive node. These guys are destined to become the heart and its vessels. They stream forward. This is the third wave that moves forward. They're converted to mesenchymal cells, just like the story when we talked about gastrulation and formation of the mesoderm. They stay lateral to the nodal cord, though. They can't push right through the nodal cord. And they go around the precordial plate. When they arrive cranially, they finally stop on the cranial side of the precordial plate, and they start to conglomerate or group together, and this group of cells is called the cardiogenic mesoderm. Why? Because they're mesenchymal cells, they're mesoderm cells, and they all form above or anterior to or cranial to the precordial plate. And it turns out they give rise to the heart and the great vessels like the ascending aorta, the aortic arch, the great vessels, superior vena cava, inferior vena cava, pulmonary trunk. There's the great vessels right there. They're kind of horseshoe shaped, so that some authors give them the a.k.a. the cardiac crescent. So cardiac mesoderm, cardiac area, cardiac crescent. First heart field's another a.k.a. Those are all a.k.a.s for this cardiogenic area. Moore's Chiropractic Board of Examiner book, so call it cardiogenic area. Right? Let's look at a picture sometime is best. So here's our kind of dorsal to ventral view. We're seeing structures, they're kind of blurred because we're seeing this is the ectoderm above, we're seeing underneath the ectoderm here. Nodal cord is right underneath the ectoderm, precaudal plate underneath the ectoderm. And then here's the cardiogenic area. So it forms a horseshoe shape outside. So this is going to form into the heart and great vessels. Everybody good with that? What forms what? So latest research says a little bit more specific on a cardiogenic area. Cells passing through the cranial region of the primitive streak go on to form the outflow tracts. So the pulmonary trunk, ascending aortic arch are the outflow tracts. And cells passing through the middle of the primitive streak turn out to form the actual ventricles. And cells passing through the caudal region of the primitive streak form the atria, right and left atria. So we actually know, and we could take a picture of that. Here's that same dorsal to ventral view, which would, dorsal to ventral is, this would really be a coronal view. But here's the primitive streak, which is on the back here of the, we're not looking in between, we're looking at the ectoderm here. There's a primitive node here. We made the nodal cord. That would be in the next layer down, and all of these would be, this is all occurring in the mesoderm layer. But this first wave of what's going to form the outflow tract, second wave of cells are destined to form the ventricles. Or the third, I shouldn't say wave, but the cells coming from, this is all the third wave, right? The third wave of cells. But the ones that come from the more anterior region form the great vessels. The middle region of the streak, they form up, they're forming that horseshoe up here, right? But these ones are destined to form the ventricles. And the ones from the dorsal part, or I'm sorry, the ones from the caudal part of the streak, they are going to form the atria. Okay, we know that from research. Okay, another structure, even further, this is the furthest structure, furthest cranial than other structure. This is the fourth wave of anteriorly migrating mesodermal cells that we need to talk about. And they form something called the septum transversum. Huge mess. The authors are all over the place. Some don't even mention it. Most authors fail to even mention the development in this important area. Larson goes on a rant about this. But at the beginning, week 3 is over. Beginning of week 4, about day 22, this forms. Fourth waves of mesenchymal cells. Epiblast cells migrate through the primitive streak like before. They head forward. They head toward the cranial region. They pass the precaudal plate. They pass the cardiogenic area, and then they pile up all the way at the very end, most cranial part of the growing embryo here. They pile up and they form a thick bar of mesenchymal tissue, or mesodermal tissue, if you will, and that is given the name septum transversus. So here's our primitive streak at this point. It's almost, remember, it's going backwards and regressing, almost gone. Nodal cord is full power here. Neural plate hasn't, should already be starting to form. It's not shown here. Precaudal plate, third wave of migration formed the cardiac region, cardiac area. Now the fourth wave is going to form this thick band of tissue up here called the septum transversum. Who cares about that thing? We're going to see, right now we're going to see, will form the separation between what becomes the thoracic cavity and the abdominal cavity. Again, second quarter stuff, but you got to know it now. Therefore, it goes on to form the diaphragm, and that's exactly the bottom line here. The septum transversum forms the diaphragm. It also, strangely, it forms the liver as well. How does it form the liver? We know BMP and Wnt genes are activated on the right side, cells on the right side, which are released, and it morphs into the primordial liver. Okay, so really the liver forms within the diaphragm, you could say. We'll get to that when we get to that. All right, so that's the end of of mesoderm differentiation. I think I'm going to stop it right here, and we'll pick up on somitogenesis in the next video, which is super important. We'll see you in the next video.